Okay, good morning and hope you guys had a great weekend. Today we are going to look at microbial growth and also measure microbial growth. So there are two subtopics we're going to be exploring today, microbial growth and measurement of microbial growth. So I've put a small outline for us to appreciate how we run through. We'll define microbial growth. We'll look deeply at the bacterial growth cycle and understand the microbial growth curve. And it's indicated to also measure microbial growth. We'll look at the different techniques that are employed by microbiologists to measure microbial growth. Of course, the last topic is not for today. Factors affecting microbial growth. That will be the topic for the next class. So let's begin with some definition. When we refer to growth in multicellular organisms, we are always usually looking at an increase in size. When you look at a baby today, 5 years after you look at that same baby, he or she is already too grown up. You yourself can reflect your size about 10 years back. You realize that you have actually grown. But in microorganisms, when we refer to growth, we don't look at an increase in size, but we look generally at an increase in cell number. So because they are too small for us to observe a change, a noticeable change in size, we instead measure microbial growth by counting the cell number. So when a microorganism divides, we usually say it has grown. So when we measure microbial growth, you understand that better. When an organism divides over and over, in microbiology, we say the organism is growing. That is what microbial growth is, an increase in cell number. We must note that in some multicellular, multinucleated organisms, chromosomal replication is not accompanied by cell division, which means that the cells are still together, but they have an increase in the number of nuclei. In microorganisms, an increase in chromosome number is followed directly by a division in chromosomal number, and that's what results in the growth that we observe. So we're going to see the different techniques that we can use to follow increase in number and the different stages that a typical organism will follow when it is in a closed culture system. I'm sure you must have heard about a cell cycle where you move from the G1 phase to the S phase to the G2 phase and then to the M phase. That is a typical cell cycle in multinucleated eukaryotic organisms. In microorganisms, we have a different cell cycle formation. So the complete sequence of events that occur from division through the next division is what we'll be referring to as a cell cycle here. Immediately a cell divides, the sequence of events that occur in that cell until it divides again constitute what we call a bacterial cycle or a microbial growth cycle, a microbial cycle. This could occur through different ways. Most filamentous bacteria will either increase in size, that is cell number, by budding, where an outgrowth occurs on the main mother cell, and then they develops into a new cell. So I might instead form a spore, and this spore uh, germinates to a new organism. Some may just fragment a portion of it falls off, and this fallen portion germinates into a new organism. I must know that these are not common, all these three are not common growth techniques employed by most traditional bacteria. Most bacteria don't use these techniques. As I indicated, they are mostly filamentous bacteria with different adaptations. The typical growth that occur in a bacteria is what we refer to as binary fission. So generally, when we're referring to microbial growth, what is in the mind of microbiologists is growth by binary fission. What then is binary fission? Here, the bacterial cell starts by elongating in size. So this is the parent cell. When it decides to divide, it elongates in size. The next thing is followed by a duplication in DNA molecule. So the new DNA molecule duplicates itself and then separates into two. A cross wall called a septum builds across the two different nuclei and then finally separates the organism into two different progeny cells. This is a typical scenario we refer to as binary fission. And as I indicated, it is the most commonly employed technique in division by most bacteria. Let's look at microbial growth now itself, microbial growth curve. As I indicated, when we allow microorganisms to grow in a closed system, a pattern of growth has been observed consistently with most of these organisms. I said a closed system, I'm referring to a system where at the beginning, we supply the organism with every growth requirement and we close the system. So by closing, I mean that we don't add more nutrients, we don't remove waste, we don't add oxygen. So we just observe it grow within that constant environment. Observe the changes that occur in the organism within that constant environment. So 4 stages have been well identified in what we describe as a microbial growth curve. 4 distinct stages have been identified. The first is what we call a lag phase. A lag phase. During a lag phase, as you can see, the microbial concentration, the microbial cell number is constant at the beginning, and then soon it starts increasing. It is constant. You see that at the beginning, the line is fairly straight, and then soon it starts going up. So this cell number starts increasing, meaning that at the very start, the cell is not dividing. At the very start, the cell is not dividing. Then suddenly, the cell begins to divide. So I'm sure you have your notes in front of you there. I'm going to use this curve to describe all the events. So I'm not going to be flipping the slides. So I expect that if you have your note, you want to follow, it's fine, but I'll prefer you give me your attention and let me explain it directly from here without flipping the slides. Again, don't forget to call my attention if I need to explain something again. Don't forget to halt me and call my attention to that. And always remember to keep your microphone muted so that I can concentrate. So as I was saying, at the very beginning, the cells are not dividing. Why? Microbiologists assume that these organisms, these cells have just been introduced into a new medium. They have just been introduced into a new fresh medium. So some of them might have been wounded. Wounded cells will need to regenerate themselves before they can start dividing. Some might have been wounded. Wounded cells regenerate themselves before they can start dividing. Another reason for the stationary phase is that this medium into which we have just introduced these organisms might be a different medium from where the organism was previously growing. That will mean that the organism will need to synthesize new what? Enzymes to be able to metabolize a new medium. Since it was growing in a different medium, it had different enzymes to metabolize that medium. Now suddenly it finds itself in a new medium. So it will need time to synthesize enzymes that will be able to metabolize this new medium. The third reason is that since the organisms have just been introduced into a culture medium, they will need the necessary enzymes to start growth. So they will need to synthesize new messenger RNA. They will need to do transcription. They will need to now do duplicate themselves. So duplication usually requires certain enzymes. Cell division requires certain enzymes to facilitate that. Of course, we know of DNA polymerase, we know of helicase, and so on. So these enzymes need to be synthesized to prepare the organism for growth. So all of this takes time. That is why, again, at the beginning, the line is fairly straight. And then, with time, these enzymes have been synthesized, whether for growth, whether for division, or for synthesis of the new, or the metabolism of the new medium, or simply repairing of the injured cells. This will take time, and at one point, this is done with. And when it is done with, the cells gradually begin to divide. And at optimum conditions, enough enzymes present, cells viable, the cell could enter what we call a logarithmic phase. Again, the length of this lag phase depends on other factors. For instance, the age of the cells. If the cells colony, the batch from which we are trying to reactivate is too old, it will take a longer time for it to repair these injured cells, synthesize these enzymes before division begins in earnest. So it takes a longer time. But if it is a very young, fresh colony, it is going to take a very short time and then it picks up division. So when it finally picks up, whether at what time, it grows at what we call optimum logarithmic rate, given its growth potential. Some organisms usually take about 15 minutes, some 20, some 30, but in rare cases, we could have up to 2 hours. And in very rare cases like Microbacterium tuberculosis, it could take even up to 2 weeks. But generally, it's a matter of minutes. The number of cells in double, the number of cells double, the number of cells double. What do I mean here? That for every cell, we have a doubling number or a doubling increase in number. And within the time interval, we can now multiply that as a logarithmic factor of increase. So a typical example is if you had just 5 cells at this stage. Here we have 1, 2, 3, 4, 5, 6, 7, 8. Let's say we have 8 cells at the lag phase. When we enter the logarithmic phase, these 8 cells are going to double. For instance, if the generation time, the time within which it takes to complete a division cycle is 20 minutes, it will mean that after 20 minutes, we are going to have 16 cells. After another 20 minutes, we are going to have 32 cells. After another 20 minutes, we are going to have 64 cells. you can keep the arithmetic going, and you realize that after 1 day, we should be having 1 million or more. So that's why it quickly gets too turbid. Because this stability area from here to here is an indication of how much concentration of cells we have after that time interval, let's say 3 hours, compared to how many we had at the first hour due to the factors I just enumerated. So the rate of increase, as I indicated, depends on the generation time of the organism. If the generation time is 15 minutes, it will mean that after 3 hours, we might be having something like this. If the generation time is 1 hour, it means that after 2 hours, we might be having about 16 cells. If the generation time is 2 weeks, it will mean that we might not observe any change in 8 hours. But typically, as indicated, microorganisms have a very short generation time. Again, it's a factor that we'll be exploring. Have a very short generation time, usually minutes. So within a day, we should have a huge population of cells to deal with. Now, at one point, things don't get rosy forever. We had a fixed nutrient concentration that we supplied the organism. The nutrient was fixed, and as this number is rapidly increasing, it means that what? They are utilizing the medium. The nutrient that we supplied are being used up. If the organism were aerobic, they are also utilizing the oxygen concentration that was found in this tube at the beginning. So they are actively depleting the oxygen concentration. And of course, when you grow, you release waste. So waste products are accumulating, and some could be toxic to the cells themselves. So at one point, growth is halted. At this stage, called the stationary phase of the growth cycle, the rate of division equals the rate of death. So some cells begin to die because of these factors I just enumerated. They don't have enough nutrients to compete to eat. So those which could compete faster and better continue dividing, while the organisms that can't compete die. Toxic waste accumulate. Organisms that are more susceptible to this toxic waste will die, while those which could accommodate a toxic environment survive. And I must note that a lot of metabolic changes takes place in the cell between the stationary phase and decline phase, which we are going to discuss certainly. At this position, which we call the ideal phase of the growth cycle, the first metabolic change that takes place in some organisms is that they switch from producing primary metabolites to producing secondary metabolites. Secondary metabolites are particles or molecules like antibiotics and so on, which are used by organisms to inhibit the growth of others. So some organisms which have the ability to produce secondary metabolites, like antibiotics, start producing these antibiotics with the hope of inhibiting the growth of other organisms to limit the competition for available oxygen, available nutrients, and available space. Of course, some organisms are still dividing, which means that nutrients are still being used up, oxygen is still being depleted, and waste are still accumulating. So at one point, the rate of division is exceeded by the rate of death, and the cell cycle enters what we call a decline phase. As I indicated, we have more organisms dying than they are dividing. Again, the reasons I highlighted in the stationary phase are still continuing here. We still have oxygen continuously dropping, concentration continuously dropping, nutrient concentration continuously dropping, waste accumulation continuously increasing, and only a very few organisms can adapt and survive these conditions. So we'll have very few living cells, while most of the cells, some microbiologists hypothesize that some of these cells actually entered a non-viable but controlled a viable but non-cultural state in which the organisms are alive but not dividing. So they simply stop dividing and enter a more a comatose state, if I must use that, which permits them to survive but not divide because division conditions are harsh. And I must know that these are generally permissible conditions in the environment, in the natural habitat of organisms. And when we're doing culture, we realize that most environmental organisms exist as these viable but not culturable source conditions. This can remain like this for forever. I mean, some source will remain viable for a very long time. They are not dividing, so they are not using any nutrient. They don't need any. They can, so they will remain for a very long time viable. But dividing source at one point will cease to divide if the conditions continue to get worse. So when we are doing a microbial analysis, if we are counting viable cells, then this line will never get to the horizontal line. But if you are counting dividing cells, actively dividing cells, then this line might get to the horizontal line. So I've already explained all these changes at the different phases of the growth circle. So except there's a question, if there's no question, the kinematics of microbial growth, I left it out for you because it is beyond the scope of your course. I don't have enough time to cover as many topics as I would have loved to. So I left out the kinematics, but feel free to read on the kinematics of microbial growth. But I gave an example already by taking the 8 cells and multiplying to 16 and so on with doubling time. That's what the kinematics is all about. So with that, we'll quickly move to the second part of the lecture, which is measuring microbial growth. Except there's a question to explain on the first. Please, you cannot text me. I can't read text, and I cannot be lecturing at the same time read in text. I've said if you have an issue, simply unmute yourself and ask directly. I'm not a magician. I cannot be trying to give a lecture and at the same time I'm reading text in the chat forum. It can't work. So as I indicated, if you have a question, you could keep it down and we'll be able to answer. If you have dropped a text, I'll be able to read the text at the end of the recording. But if the thing is urgent, of course, always unmute yourself and interrupt the lecture so that we could get it cleared. That said, we move to measurement of microbial growth. Now, what do we mean by measurement? Yes, go on.
So we have done microbial.
Hello?
Yes, I'm saying that we have done microbial growth curves in another course, but there is a phase there that is like deceleration phase, which is before the stationary phase, and I don't know if you can talk about that. It's different for some books or what, I don't understand.
A deceleration phase?
Yes, sir.
Maybe it's another way of calling it. We have a decline or a death phase. We have just 4 phases in the microbial growth cycle. To the
but
best
that
of my
phase, sir.
Hello? Yes. Please go on.
Okay, sir, but even if there is a decline phase, it should be before the stationary phase, because the manner was explaining like after the lock phase.
No, it can't be possible. It's not possible. I'll come back to that at the end of the class, but it's not possible. That's my comment on that for now. Okay?
Okay, sir. Thank you.
I'll come back to it at the end of the class, but we have just 4 phases, and there's no way a decreased phase should come after the stationary phase. As you saw me explaining, I was giving reasons why a phase sets in. So be reflecting on that while we'll come back to it, okay? Good. Measurement of microbial growth. So there are so many different ways we want to measure microbial growth. It depends on the objective of our assay, why we want to measure. some will directly measure viable cells. So it takes into account only cells that are actively reproducing and still alive and actively reproducing in some cases. Remember, I just mentioned that in some cases we might have viable cells, but which cannot be cultured. So if your viable assay is culture based, you should have this issue of viable but non-culturable cells in mind. In some cases, we just want to have a total number of cells present. In some cases, we want to indirectly measure the number of cells present or the concentration of cells present by either measuring the mass, measuring the cellular organelles, or even measuring the products of metabolism. So we're going to look at all of this. But the most popular uh, method used is direct count. Okay, direct count of cells. So here we use a specially designed slide. A specially designed slide, we apply the sample onto a specially designed slide and we'll observe under the microscope. We'll see the cells under the microscope appear as you see on this grid here. So from there, you can take the time and count the number of cells that are present and report. There's a specially designed grid that is being used for this called the Petro-Hauser counting chamber. So I'm not sure we'll be able to do this particular one in the laboratory, but those who have been able to witness blood counting is a very blood cell counting in medical laboratories is a very similar technique. You apply the sample, maybe it's a liquid sample or stool sample or contaminated water. You apply it here and then in some cases we might stain, but mostly we try to count the cells with the use of a magnifying microscope to report. Now, when we count the cells, we have to report it by dividing the number of cells counted by the volume of the area counted. So there's a more under elaborated formula for this. It is not of interest in your scope, but what you need to know is you use a specially designed grid, the Peters-Hauser counting chamber. You apply the sample on it. You take the grid under the microscope, and then you count the cells, and you report by dividing the number of counted cells by the volume of the area counted. As I indicated, there's a way to get this volume. We can also use electronic counters to measure microbial growth. There are two popular counters that are being used now. We have the culture counter and the flow cytometer. In the culture counter, the microbial suspension is forced through a small hole, and the flow of electric current is obstructed by the presence of these microbial cells in that small hole. So we have a small hole that is linking the anode and the cathode. And electric current is supposed to flow through that hole from the anode to the cathode. Now, when we introduce these cells to the sample we want to measure to this hole, we are assuming that microbial cells will obstruct current flow. So whenever there's an obstruction in current flow, we count a cell. Whenever there's an obstruction in current flow, we count a cell. Whenever there's resistance in current flow, we count the cells. So that will give an idea of how many cells are present in our sample. The flow cytometer or the flow cytometry method is also a very good method, but more advanced, and it's being used in medical studies to count so many other cell components, even in the body, not only in microorganisms. Then we have the membrane filtration method. We must note that most microbiological analysis are within a very scarce environment. That is, the number of microorganisms present in our samples are actually very, very few. So we may have to always filter a huge sample before we can have a measurable or appreciable quantity of microbial cells. So in this case, if you want to analyze maybe drinking water for microbial contamination, you might have to culture about 50 liters of water to have growth on a slide like this. So in this case, it becomes very difficult for you to culture 50 liters of water. So what microbiologists simply do is they filter that sample. They'll filter that 50 liters through a membrane that is capable of trapping bacteria, has pores small enough to trap bacteria. So the pore size of the membrane is usually 0.45 micrometer. With this size, you are capable of trapping even the most smaller or the smallest uh, bacteria. So when you filter the 50 liters of water, you take the membrane, you take the membrane onto a culture medium. You take the membrane into a suitable culture medium and you incubate for 24 hours. After incubation, you are going to observe colony growing where they were trapped, where the individual cells were trapped. You're going to see them appear now as colonies. A single cell is going to divide within that 24 hours and increase in number and grow, and therefore we can now be able to observe the growth for a grown cell, and we can even count this number of cells. Again, note that if the cell is unable to grow, we will not be able to see it. So this only counts cells that are capable of growing on this media. Because if the problem could be the media that is too selective for the organisms. So generally we'll use a general purpose media in this analysis. I'll be talking about that again during culture. We use general purpose media in this analysis so that we give the room for almost every organism to grow. But as you'll be understanding during culture, not all organisms can grow on every media. There are always some that will not be able to grow. And again, we also learned of viable but non-culturable cells. So these VPNS cells, VNC cells are generally difficult to culture, and it becomes difficult for us to ascertain their presence in our sample. But of course, with a more suitable environment like within the human system, these viable but non-culturable cells are able to grow again, which means that they can still cause an infection. Now, we move to more sophisticated viable methods that are commonly being used. And here we are simply using the plate culture method. It can either be spread plate technique or pour plate technique. We'll be able to use these two techniques in the laboratory, spread plate and pour plate, during our practicals. Here we are usually interested, unlike in the filtration method, we are interested in more contaminated samples. We're interested in more contaminated samples. So there's no need to filter because there's already enough to grow. So what do we do here? We might instead need to dilute the sample, because if we culture, for instance, a fecally polluted water sample, if we culture it directly, we are going to observe enormous growth that will be difficult for us to count and measure the number of cells or report the number of cells that are present in that sample. So what we usually do is we do what we call serial dilution. To do a serial dilution, again, I'll be elaborating on this technique in the laboratory, and I'll take you through the process of serial dilution in the laboratory. But remember that we dilute to reduce the concentration of cells. Generally, for microbiologists, if you culture for enumeration and the number of cells are more than 300 on a plate, we report it as too numerous to count. We assume that it will be difficult for you to count 300 cells, more than 300 cells accurately and report. And besides, the number of cells being 300 will already mean that there are some growing on top of others. So you cannot again enumerate as colony forming units as we typically report. So it is too numerous to count. And of course, in a numerical enumeration assay again, if there are less than 30, we report as too few to count. So the ideal number for enumeration for a diluted sample is between 30 to 300 cells. I think I've integrated that in your notes. Between 30 to 300 cells, that's the ideal number to count. So when we count, when we dilute, we could have different dilution factors. One, 1 in 10, 1 in 100, 1 in 1,000. So we'll look for the dilution factor that will have cells between 30 and 300. And within that dilution factor, we count the number of cells that have grown. And then our assumption is that each colony that we are counting is arising from an individual cell that was found in our sample. So if we have 50 colonies counted, for instance, we are assuming that there were 50 cells in the quantity of sample that was inoculated onto that sample and incubated, and therefore we report it as 50 colony forming units per mil. We don't report it as 50 cells. The reason is that we are just assuming that a single cell grew into a colony. That assumption could be wrong because there could be two cells paired together, and those two cells divided into a single colony will miscount in that case. There could be two cells close to each other, and then they grow and merge into a single colony, will be also miscounting in that assay. So the most appropriate way to accept your level of error is to report but as colony forming units per mil, CPU per mil. So we can also measure the mass of the cells that we have. This is usually applicable in filamentous bacteria and algae and fungi. So what we'll do is we collect the sample one to enumerate. We wash, dry, centrifuge. We centrifuge, wash, dry, and then we measure the weight of what we have, and then we can now extrapolate that to an approximation of the number of cells that are present in our sample. Spectrophotometry is another method very close to electric method, but the difference here is that we are basing on the assumption that microbial cells are capable of scattering light. So when light falls on is impacted on microbial cells, they scatter, they diffract the light to different angles, and therefore the amount of light that is transmitted is not the same as the amount of light that was incident on the sample. So with the use of a spectrophotometer, we introduce the amount of material that we want to, the sample that we want to enumerate, the number of cells within that sample. The more the cells we have in our sample, the more the scattering of the light. So I'm saying that the more turbid our sample is, turbidity is directly proportional to the number of cells or the level of contamination of our sample. So the more turbid our sample, the more scattering the light will be done or we experienced in the spectrophotometry, and the lesser transmitted the amount of light we'll be able to dictate. So we can either measure the difference between incidence and transmitted light, which will be a proportion of the amount of light absorbed or scattered by our cells, or we could directly measure absorbance and translate it to the amount of cells. So we can measure percentage transmittance, or we can measure absorbance to determine the density of our cells, which is directly proportional to the number of cells that are present in our sample. Finally, I indicated that we could also measure cellular substances. These cellular substances could be organelles or could be metabolic products. Okay, some organelles are generally constant during the growth cycle, and these are the type of organelles that we want to measure, not organelles like vacuoles that will disappear at one point, will appear at one point. If you measure vacuoles, then it will not give an idea. At one point, the cell could be highly vacuolated, depending on whether it's growing in a hypotonic or a hypertonic solution. And therefore, we cannot be able to measure that. But if we measure ATP, for instance, if we measure, if we measure the amount of nitrogen, which is found in almost all nuclear material. If we measure the amount of ATP still, which is a metabolic product, if we measure the amount of protons, we can go to translate this to the concentration of cells present and then have an idea of the number of cells that are present in our sample. So the higher the concentration of ATP, the higher the number of cells producing that ATP. The higher the concentration of protons, the higher the number of cells producing those protons. So that is the assumption. There again, it's just an assumption and it might not be very accurate in certain settings. And that is why, of course, we have different measurement techniques, because each technique, as already been identified to you, has its limitations. Again, I fail to point out that in this spectrophotometric technique, we are not sure that everything that is capturing light here is a microbial cell. We could have contaminants in our medium which are scattering light, and we're assuming that they are microbial source. That also applies for the electric quota counter and flow cytometry. Not everything, the obstruction, the resistance to current flow is not necessarily because of a microbial cell. Every contaminant that is passing across that pore will also cause resistance and will also be detected as a cell, which will lead to overestimation. Again, not all of these methods can be able to measure viable cells except culture-based methods. So we are not able to differentiate dead cells from living cells from all these methods except culture-based method. Again, culture-based method also had the disadvantage of what? Being able to identify only culturable organisms. Viable but not culturable organisms could not be identified. So I think that is it for the first segment of this lecture. I will stop the recording and stop sharing my screen.